First of all, I would like to thank the organizers for giving me a chance to present my work over here. So I'm from IIT Goa. I'll be talking about limitations to measurements in cellular systems. Now, the thing is, as far as cells go, right, evolution has made them into these very good information processing machines. For example, if you're looking at this bacteria E. coli, it can detect 3.2 nanometers of this attractant aspartate. What is so interesting about this is that this corresponds to only 3 attractant particles in the volume of the cell. That is what this concentration is. So, this is the level. I mean, first of all, because these sizes are very small, you're having a lot of these fluctuations, these noise fluctuations, fluctuations because of receptors binding, unbinding, as well as the reactions happening within the cell. But even with that, it can detect these kinds of concentrations which are corresponding to 3 attractant particles in the volume of the cell. If you're looking at eukaryotic organisms, that are the organisms with a nucleus, a very common organism which is studied in biology is this dictostelium. And then over here also, what is seen is that they can detect these CAM concentrations of these magnitudes. But these organisms, they basically detect concentration gradients because of their size, because they're like a few micrometer sizes. And then what is seen is that altogether, on an average, there are like about 4,000 CAMP receptors on the dictostelium. And in case you're having a concentration such that concentration of the CAMP such that about 200 receptors are occupied, even when the difference in the front and back of the receptors is only 5 receptors per cell, even in that particular case, this particular amoeba can basically detect that there is a signal and it can move in the direction of the signal. Okay. So despite the noise involved in the measurement, and by noise involved in the measurement is this noisy chemical reactions happening inside the cell, or the the binding kinetics, the noisy binding kinetics of the ligands to the receptors on the cell surface, it still can detect these kind of gradients. So what I'm going to be talking about is trying to understand two systems over here and two problems. One problem that I'll try to talk about is the limitations to gradient measurements in dictostelium. And the other problem that I'll be looking up at is the limitations to glutamate, which is a neurotransmitter, concentration measurement in neuronal signaling. So let me get to the first part of my talk. This is based on a part of the work in this paper. And yeah, so now what was done was, like in this 1982, they did this experiment where what they did is, for this amoeba, they hit it with these pulses of CAMP, okay, through this pipette. And what they saw is that the amoeba cannot, this dictostelium cannot detect pulses shorter than 5 seconds. Okay. So now let us try to understand this, try to understand this model using some physics. I mean, trying to understand this thing happening through some physics. So what I've done is, the dictostelium that is there, this is the pipette, it is giving out these CAMP particles. I just try to idealize the system as a sphere. There's the source, okay, which is giving out these particles. You're assuming that there are receptors located on the sphere at each angle theta, and then the probability of occupancy of the receptor, okay, can be written as this particular attachment rate times the probability that the receptor is not occupied, and then the probability of detachment. Okay, and then what you have is that this current, okay, of the CAMP, okay, is basically equalizing, is the one which is responsible for attachment of the receptors of the CAMP to the receptors. So that is taking care of this relationship. Now, see, my main problem over here is to look at the limitations that are happening as far as measurement by the cell goes. So let me give the cell a benefit of doubt, and let me say that it has the best readability obviously is when the cell has equilibrated with the CAMP concentration. So let me just consider, okay, the cell has equilibrated with the CAMP concentration, because it makes solving of the problem even a little bit more easier. Okay, in that particular case, you will just have this boundary condition. If you do that, then what you have is, this is the source, this is the diffusion equation with the source located here. You're having this boundary condition, you can solve this, and you can get a concentration profile. Now, details I cannot go in this small talk, okay, but the error in the measurement of the concentration, okay, basically goes as this particular formula which is derived in the paper, and this is related to the variance in the receptor occupancy, okay. And then in order to discern a gradient, what you would be wanting is that the error that is the concentration that is there, the concentration difference between this point and this point should be greater, at least greater than the error that is measured at this point versus this plus this point. Okay, this is just a normal condition. In that particular case, you'll be able to measure the gradient. Okay. Now with that thing, if you like do a little bit of more simplifications, you get this particular equation that the time of measurement should at least be greater than this factor in order for the dictostelium to basically detect the signal. And the smallest value this thing takes is when A is equal to R. A is the, okay. And then what you get is that this is the equation that you get for the T smallest. Okay. The thing is, as far as these rate constants go, okay, these have been measured. I don't have the citation for the paper, but in the anterior pseudopod region, okay, that is the region facing the chemoattractant, the rate constants are of this value. In the posterior tail region, on an average, these are the rate constants. And in case you consider some kind of an average value between these, as your K minus, what you see is that the smallest time that the dictostelium can basically, the shortest pulse, at least from these calculations, that the dictostelium can measure is 4 seconds. And what you see is that in the experiments, only pulses larger than 5 seconds can be measured. Okay. So see over here, what we have done is one thing. Okay, what we have done is we have only looked at some receptor dynamics, and we have come up with a answer for the smallest pulse, a pulse of the smallest size you can measure. And actually, dictostelium that is there is operating around that time scale. So it has basically evolved, okay, to make sure that whatever noise related effects that are there, okay, are, even though there are noise related effects, the system has basically optimized itself that it can detect the shortest possible pulses which are physically possible. Okay, that is the punchline of this particular portion of the talk. Oh god. Okay, so this next talk, this next portion of the talk, I'll be talking about limitations to glutamate concentration measurement in neuronal signaling. This work has been done in collaboration with my PhD student Swayam, and I would like to acknowledge this EEQ grant from SERB. Now, as far as I have like 2 minutes left. Oh, okay. So as far as memory formation goes, okay, there's a theory of memory formation. Okay, this is what I've read in the literature. This is the possible theory in which memories are formed. When you're learning something, right? When you're seriously studying something, you basically, the neuronal, the action potential that is there, is basically going to be a high frequency action potential. When you're not seriously studying something, it's going to be a low frequency action potential. What happens is that when an action potential basically reaches the synapse, you have this opening up of these vesicles, okay, on the surface of the and then what you have is the emission of this glutamate. Okay, this glutamate, what it does is there are these two kind of receptors, NMDAR, AMP receptors. They will lead to the influx of sodium ions, but it's only when high frequency signal is coming in through the action potential that is corresponding to actually learning something. The concentration is so high that you will basically have more amount of sodium coming in. That is going to start the influx of calcium through these NMDAR receptors. Those will then lead to phosphorylation of proteins, etc., and that will lead to the entry of these AMP receptors, more AMP receptors on the surface of the other synapse, and that will basically lead to, like in the next case, once you're having more receptors over here, next time when you're having a low frequency signal, even in that particular case, this thing can get activated. So at least what you see is that some degree of glutamate concentration readout accuracy is needed, okay, in order, as far as long-term potentiation goes, because you don't want a very light signal, just in the beginning, if you just had a very light signal to like act like these receptors are seeing it as a very strong signal. Okay, so then the question was, okay, what is the limitations? Can we basically try to see how these noise-related effects lead to noise-related effects affect the readout, okay, of this glutamate signaling? As far as the signaling goes, I just have 15 seconds left altogether. After that, 2 minutes. Okay, yeah. So then, as far as the signaling goes, the glutamate signaling goes, this NMDR receptor, okay, the way it gets activated is, what you have is that it gets activated when two glutamate ligands and two glycine ligands are attached. Once that happens, what you have is this calcium gets inside the cell, and then what it will start doing is it will basically go and activate this kinase, and the kinase only gets activated when 4 calciums are basically attached to it. That thing will lead to activity which was like this AMP receptors coming and like hitting back on this membrane. Okay. So now let us try to understand this particular problem. In case I write down the equations, this is how they look like. Let us not go over it. The main thing is, if you just look at the concentration, if you just look at the attachment rates, what you see is that in principle you would just have like full occupancy. That just on a naive guess. But what actually happens is that the glutamate that is there diffuses very fast. Because the glutamate diffuses very fast, there are these calculations done in this paper which suggest that the average occupancy is like 30 to 81%. So what we considered is, okay, once we do a calculation, then we can get a renormalized value of these attachment rates. And in principle, one can do these Monte Carlo simulations to basically see how the activity is going to look like. Might be I can just, okay. This particular portion was just trying to say that, I could try to do some analytical calculations over here, but let me just skip over that because of the lack of time. But if I just, once I do the Monte Carlo simulation and the analytical calculation that I do, I can basically get a very good agreement, and I can actually get a result which agrees, I can get a nice form for the results for smaller values of K plus CT. Let me just go over that. But the main thing is, for this renormalized value of K plus KCT, if I evaluate, okay, the error in the concentration measurement just for one receptor, I'm getting these values. But if you're having n number of receptors, then it is going to be scaled by square root of n receptors. And what I see is that if I actually evaluate DC by C, what I see is that the error is like about 50% to like 10%. Okay. So what it tells you is that the measurement of these receptors can actually at least tell you whether the concentration of the glutamate was strong or weak. It won't basically, at least that much of this, just the number that have been chosen, it's just 400. That can at least tell you whether the concentration that was there of the glutamate was strong or weak. So in some sense, evolution has basically chosen this number of receptors. There might be various other reasons, but this thing becomes a very strong reason because the number of receptors chosen is just 400, not 4,000 or 40. As far the order of magnitude goes, because it is telling you that, at least with this number of receptors, you will be able to discern what the concentration is, whether the concentration is strong or weak. Yeah, sure. This is my punchline. Yeah, thanks.
Okay, let us thank the speaker.